Assalamu alaikum dear students. Today we will be discussing about the synthesis of the hemoglobin. Hemoglobin first appears during the pro-erythroblastic phase of the RBC production. At that time, the cell has all the organelles including the mitochondria along with the DNA, that is the nucleus. So, succinyl CoA is formed during the Krebs cycle, and two succinyl CoA combine with two glycine molecules, which is an amino acid, and hence form a pyrrole ring. Now, this pyrrole ring combines with a combination of four pyrrole rings takes place and helps in the formation of protoporphyrin 9. Now this protoporphyrin 9 gets combined with a ferrous ion, that is Fe2 plus ion, which causes the production of a heme molecule. So the protoporphyrin 9 combines with a ferrous ion, causes the production of a heme molecule. Now the heme molecule combines with a polypeptide chain, and depending on the type of the chain that combines with it, a hemoglobin chain is formed. This hemoglobin chain in the adults can be of two major types. One is the alpha chain and the other one is the beta chain. Whether it is the alpha or the beta chain, it depends on the polypeptide chain that has combined with the heme molecule. So in an adult hemoglobin molecule, two alpha and two beta chains combine together and give rise to a hemoglobin A1. So that was it for the synthesis. Let us just revise it, that two succinyl CoA plus two glycine form a pyrrole ring, and four pyrrole rings then combine to form a protoporphyrin 9, which combines with a ferrous ion forming a heme molecule, which combines with a polypeptide chain and giving a hemoglobin chain, and two alpha and two beta chains combine together to form a hemoglobin A1. Types of hemoglobin present during the fetal life. Hemoglobin F is the one that is present at the later stages of fetal life, and it comprises of two alpha and two gamma chains, while Goover 1 and Goover 2 are the ones that are present during the embryonic stages. The Goover 1 comprises of two zeta chains and two epsilon chains, while Goover 2 comprises of two alpha and two epsilon chains. Now comes the adult hemoglobin. The primary type of adult hemoglobin is known as HbA1, which comprises of two alpha and two beta chains, while a very small amount of adult hemoglobin is also present in the form of hemoglobin A2, which comprises of two alpha and two delta chains. Another very important type of hemoglobin is the glycosylated hemoglobin, that is HbA1c, which is very important in a physician to determine the glucose status of a patient. Now comes the methemoglobin, in which instead of ferrous ion, the hemoglobin is attached and is formed by the ferric ion. This amount of hemoglobin is very less in quantity but is present to some extent. Carboxyhemoglobin is a very lethal form of hemoglobin in which the carbon monoxide is attached to the hemoglobin, which decreases or causes a total lack of oxygen combination with the hemoglobin. Then we have oxyhemoglobin, that is the hemoglobin is when attached to a oxygen is known as oxyhemoglobin. Now let's just very briefly talk about the hemoglobinopathies. The two very important hemoglobinopathies are sickle cell anemia, along with that there is thalassemia. First of all, sickle cell anemia is the one that is present in African ethnicity, in which the beta chain that has different amino acids present on it, then over there the sixth amino acid, which comprises of valine, is replaced by the glutamate, and hence causing hemoglobinopathy, which causes the sickling of the cells. In this, the cells attain a sickle shape, which occurs only when the hemoglobin is not attached to the oxygen, and these sickle cells then get stuck at the spleen, and hemolysis takes place of the sickle cells. Now let's talk about thalassemia, which is very common in our ethnicity. In it, there is either a deficiency or there is an absence of the alpha gene or the beta gene, which are part and parcel of the adult hemoglobin. That was it for today. I hope so you guys have understood. Thank you.